presentation is on transport mechanism, in particular transport. Classification of transport mechanism is as follows. First one is passive transport, which includes simple diffusion, facilitated diffusion, aquaporins, ion channels, ionophores. Active transport include unipodes, import, antipode, exocytosis, and endocytosis. Endocytosis in turn includes pinocytosis, phagocytosis, receptor-mediated endocytosis. Pumps include sodium, potassium, ATPase, and calcium pump. Transcytosis, and last one is the ABC. So let us try to understand only passive transport in this presentation. The characteristic which include there is no requirement of energy and occurs because of a concentration gradient. So passive transport mechanisms include the following: simple diffusion, where solutes and gaseous substance utilize this diffusion mechanism to enter into the cell and outside the cell. The gaseous substances which utilize simple diffusion include carbon dioxide, oxygen, carbon monoxide, and nitric oxide, and they always move from higher to lower concentration. No energy is required, but how this is a very slow process. The second one is facilitated diffusion. Here also there is no requirement of energy, but requires a carrier molecule in the form of integral membrane protein. This gets saturated at higher concentration, and it is inhibited by structurally similar solutes. However, this facilitated diffusion operates bidirectionally using ping pong mechanism, and also this occurs along the concentration gradient, moving from higher to lower concentration gradient. So good example for facilitated diffusion is glucose transportation through GLUT transporters. Now these GLUT transporters are distributed across the human body, and different organs have different GLUT transporter. GLUT1 is present on the surface of erythrocytes, kidney and placenta, GLUT2 in intestine, pancreas, GLUT3 in brain, GLUT4 in liver, muscle and adipose tissue, GLUT5 in testes and sperms, and GLUT7 in liver endoplasmic reticulum. So with the exception of GLUT4, all the other GLUT transporters are not dependent on insulin, except GLUT4. The glucose entry into liver, muscle and adipose tissue requires the presence of insulin circulation. The next one is aquaporins, which are integral membrane proteins, and they are mainly involved in transfer of water inside and outside the cell. So they are numbered from 0 to 9. And diabetes insipidus occur as a result of defective aquaporin 2. Diabetes insipidus is characterized by deficient production of vasopressin or antidiuretic hormone, characterized by polyuria and decreased specific gravity of urine. The next one are the ion channels, which are responsible for quick transport of electrolytes, and they have been implicated in nerve muscle propagation, synaptic transmission, and secretion of biologically important molecules molecules. There are ion channels of two types: ligand gated and voltage gated. Ligand gated channels open by binding of effectors. Good example is acetylcholine receptors, which are present on the postsynaptic nerve terminals. So from the presynaptic nerve terminal, acetylcholine is released, which goes and binds to the receptors on the postsynaptic transmission, so that the action potential propagates further, and the receptors facilitate the entry of sodium into the postsynaptic transmission, bringing about the postsynaptic transmission. Next is the voltage gated, which are opened by membrane depolarization. The examples include GABA and glycine. They open chloride channel and act as inhibitor neurotransmitter, whereas glutamate opens a calcium channel and act as excitated neurotransmitter. So variety of clinical significance is associated with this ion channels. Local anesthetic block this potassium channel, decrease the entry of potassium, and they decrease the action potential. That is how they block the pain impulse. Next is the calcium channel blockers have been used in the treatment of hypertension. This is because these antihypertensive drugs decrease the calcium entry into the smooth muscle, bring about vasodilatation, decrease the peripheral vascular resistance, and that is how they decrease the blood pressure. Next, mutation in calcium channel has been implicated in the development of cystic fibrosis, which mainly affects the exocrine glands, resulting in the production of thick viscous secretion in multiple organs like respiratory tract, resulting in leading to repeated lung infection, affecting gastrointestinal tract, also contributing to the development of pancreatic insufficiency involving the exocrine part of the pancreas. Even sweat glands are affected, where characterized by increased sweat chloride. Next one is mutation in potassium channel results in long QT interval, manifesting as ventricular repolarization in ECG, and clinically characterized by ventricular arrhythmia. There is increased activity of the ventricles of the heart. Next is mutation in sodium channel has been implicated in the development of Liddle's disease, characterized by early onset hypertension. This is because of activation of the epithelial sodium channel in the collecting tubule of the kidneys. Coming to the last passive transport mechanism is ionophore, which allow entry of specific ion into the cell by acting as channel formers. These include the fixed and the mobile ionophores. Valinomycin facilitate entry of potassium into the cell. This is the mobile, and the fixed one is gramicidin. Now, this valinomycin facilitate entry of the potassium ion into the cell instead of hydrogen ion at the level of electron transport chain, dissipating this causing uncoupling of the oxidation from phosphorylation. So it has been considered as an important uncoupler. So this was all about passive transport mechanism. So the next video will be on active transport mechanism. Thank you.